peptides, histones are captured by the wild-type strain. There is less binding of the isogenic mutant, and you can demonstrate a direct interaction between the peptide and the mutant. So the bacterial peptide, or the antibacterial peptides, whether they are cathelicidins or histones, as seen here in these electron micrographs, get captured in the hair of the bacteria before they can reach their cell wall target of action. So it's like a stiff arm of a football player preventing the defense mechanism from tackling them. Okay, the poison mushroom. So group A strep are distinguished in the clinical lab by their ability to produce a hemolytic toxin that lyses red blood cells, and this is a rapid method of presuming that you have streptococcal infection and should probably give the patient penicillin. So a number of years ago in the lab, we discovered the genetic basis for these toxins. It is a 9-gene operon that we've called the streptolysin-associated genes, and you can see here that streptolysin S, responsible for the hemolytic phenotype, is eliminated if you knock out any one of those 9 genes. And you can also clone the 9 genes into other bacteria and render them hemolytic. It's important in virulence because if you eliminate the toxin, which also causes damage to a variety of cell types, not just erythrocytes or red blood cells, you have much less necrotic injury in this severe infection that the M1 invasive flesh-eating strain causes in a mouse. If you take away the streptolysin S, it's no longer able to produce that infection. And this is an enigmatic toxin. It's never been fully purified, but because it's unstable once you remove it from the bacteria, and it's in fact directly transferred from the bacteria to the target cell. But by cloning the individual proteins and expressing them in the operon, we found that the first 4 genes in the operon, with the precursor of the toxin being gene A, are able to make this heavily post-translationally modified peptide, which then is able to produce hemolytic injury or injury in an animal upon injection. And we believe that now, knowing some of the structural elements, we can create point mutants that lose hemolytic activity and could maybe be a component of a multi-subunit streptococcal vaccine. Streptolysin O is another toxin that doesn't contribute to the hemolytic phenotype on the blood agar because it is oxygen labile, but it's very well characterized, and you can see here that it assembles in a multimeric pore to destabilize bacterial membranes. And one interesting thing we've discovered about this second toxin of strep is that it's responsible for the accelerated apoptotic cell death of immune cells. The bacteria tries to kill the immune cell before the immune cell can kill it after phagocytosis. And that's why I analogize this to a poison mushroom. May have seemed tasty at first, but then turns out to be very bad for you. If you eliminate the streptolysin O, you can eliminate this accelerated death, and you can introduce SLO into normally non-cytolytic strains of bacteria, and they are able to then kill immune cells. And you can see that this is a caspase-dependent apoptotic cell process. SLO, even before it executes the cell death program, has effects to suppress other critical neutrophil functions, including chemotaxis. What we see here is the suppression of the neutrophil oxidative burst, because if you eliminate SLO, you get a much better oxidative burst from the neutrophil. And you can see here just purified toxin causing a dose-dependent suppression of the oxidative burst of neutrophils, and we see the same thing with macrophages. And because oxidative burst is one of the stimuli that helps promote the production of neutrophil extracellular traps, Wild-type group A strep that are making a lot of this toxin can suppress the production of neutrophil extracellular traps. You see much more trap production in the isogenic mutant. So this bacteria has really completely disabled neutrophil extracellular traps as a defense mechanism because it suppresses their production in the first place, and even the ones that are made are then degraded by the DNase. So the bacteria is free to spread through the body, and that's what these patients are experiencing, unfortunately. Interestingly, if you block the caspase-dependent apoptosis triggered by the immune cells, you can promote, this is an inhibitor of apoptosis, you can promote macrophage killing of group A strep. And we found also that you can make point mutants of the SLO toxin that are very good immunogens. So this would be the normal mortality at this dose of infection. Just immunize them with an inactivated form of SLO. They make antibodies against SLO. Their white blood cells are happier, and the infection can be withstood. All right, wolf in sheep's clothing. So everybody may know this concept of a tricky wolf who is interested in eating some sheep. So he would decorate himself as a sheep and enter the flock, and the unsuspected sheep are then at risk of being captured. The capsule of group A strep is a homopolymer of hyaluronic acid, which is a non-sulfated glycosaminoglycan that is one of the most common components of our own connective tissue. So we have hyaluronic acid throughout our body, and what this bacteria is doing is basically coating itself with a human-like sugar to hide and then sneak into our bodies. And what we've discovered more recently is not just that it's hiding underneath this, but that the hyaluronic acid is able to engage receptors on immune cells and downregulate their expression to even shut down the immune system further. So this is very sinister strategy, like a wolf. So here's the capsule, especially of the blood-borne strain, which is spreading through the body after the mutation. It makes a lot of capsule. This is the structure of molecular weight of hyaluronic acid. This repeating disaccharide can extend to be 1 to 4 million Daltons in size, a huge capsule. And eliminating the capsule makes the bacteria more susceptible to immune killing, and that's been known for a long time. So what was the new discovery that I want to share with you? Well, we have been working a long time, actually, with a physician scientist in America who's from Kerala, named Ajit Varki. And he's my close colleague and laboratory neighbor. And Dr. Varki is the world's expert in a family of sugars known as sialic acid, and the way that sialic acid plays role in biology and disease and evolution. And an interesting aspect of sialic acid biology is that we have receptors on our immune cells, which are inhibitory receptors known as Siglecs, that recognize sialic acid, which is ubiquitous in our body, as a self-associated molecule, a signal of self. And when they bind sialic acid, they're going out throughout the body and they say sialic acid, self, self, self. They keep the immune cell off because you don't want to have unnecessary reactivity. And this is a major paradigm in immune cell regulation, where Siglec receptors help keep leukocytes in an off state. And our research began with a different organism we study in the lab called group B Streptococcus, a leading cause of invasive infections in newborn babies, because Ajit Varki reminded me that this bacteria has sialic acid in its capsule. And we've been studying extensively in a large number of papers how the sialic acid on the bacteria can engage these Siglec receptors and send inhibitory signals to immune activation. So that's a type of molecular mimicry of group B strep of sialic acid to engage sialic acid receptors and keep your immune cells off. So how does this come back to group A strep? Well, one day I needed a negative control for my experiments with Siglecs, and I thought, well, why don't we do group A strep? That's kind of close, at least alphabetically, to group B strep, and it has a hyaluronic acid capsule, has nothing to do with sialic acid. Well, what we found is that there was one Siglec, and it happens to be the Siglec that is most abundant on neutrophils, that when we tried to do the control experiment with hyaluronic acid, it bound strongly to hyaluronic acid. So this Siglec receptor, there's probably about 15,000 papers on Siglecs, never been reported to bind any other sugar than sialic acid. And we found that you could inhibit this by adding exogenous hyaluronic acid, but there was no cross inhibition with all the other common sulfated glycosaminoglycans in our body, like heparin sulfate, chondroitin sulfate, heparin. And remember that group A strep is making a capsule that mimics the human hyaluronic acid. And in fact, what we saw is that group A strep could also bind Siglec in a manner that was dependent on its hyaluronic acid. And then we got kind of lucky. Ajit Varki has a lot of monoclonal antibodies he's developed against the different Siglecs, and we screened them, and we found that there was one monoclonal antibody that bound to Siglec-9 and inhibited sialic acid binding, but had no effect on hyaluronic acid binding. And then a second antibody that bound to Siglec-9 and inhibited hyaluronic acid binding, but had no effect on sialic acid binding. So these were very good tools to study. And what we found was that, in fact, group A strep was able to bind to immune cells via Siglec-9 in a hyaluronic acid-dependent fashion and shut them off. So you see here that there's an oxidative burst assay for activation, and normally group A strep is able to suppress the oxidative burst, but if you prevent the hyaluronic acid engagement, you restore that oxidative burst. And there's no effect with the sialic acid binding protein. Remember that the neutrophil extracellular traps are associated with oxidative burst. You can see here, and we're using the DNase mutant so they don't get degraded right away, that group A strep, through its hyaluronic acid capsule, is able to suppress neutrophil activation and net production. If you block the hyaluronic acid interaction, then you restore that net production. And this turns out to have an effect on killing of the bacteria. So if you normalize the killing to 100% in the absence of antibodies, if you prevent group A strep from using its hyaluronic acid to bind to the neutrophil and turn it off, you get enhanced killing. And this, in fact, is what you see when you don't have a capsule. They're all easily killed. There is a murine paralog of Siglec-9 known as Siglec-E, and we found some binding of Siglec-E by the hyaluronic acid capsule of group A strep. So we wondered, if you get a Siglec-E knockout mouse that is then not susceptible to this immune suppression, would it do better than a normal mouse with respect to group A strep infection? And what we saw is that, in fact, blood from these mice is better able to kill group A strep, and that there is a reduction in bacterial counts in the absence of Siglec-9. So just think about this. The bacteria is coming up to your white blood cell and grabbing onto a receptor on its surface and turning it off like a light switch. And that's one of the reasons that this is a major pathogen. And what's even more fascinating is you have two entirely different bacteria, group A strep and group B strep, that have evolved different strategies of molecular mechanisms. One, a single 9-carbon sugar known as sialic acid. The other, a huge polymer of hyaluronic acid. They both bind to the same inhibitory receptor in its most exposed VSET domain, engage it in a functional way that sends negative signals through the cell. This involves SHIP phosphatase recruitment and counteracting of kinase cascades. So this is convergent evolution, wolf's in sheep's clothing, that inhibit immune clearance. Okay, and the final story is the canary in a coal mine. And this has to do with that protease called group A strep, and something that we've discovered that kind of rewrites the role of an important cytokine called IL-1 beta in an innate immunity. So some of you might be familiar with the cytokine IL-1. It is one of the most important pro-inflammatory cytokines in immune responses and in host defense against pathogens. And IL-1 is felt to be produced in a process that involves intramolecular assemblies known as inflammasomes. And you can see here that what happens is a number of molecules get together, most commonly a component known as NLRP3, upon sensing the bacteria or a variety of host-derived danger signals. And this is a platform for the activation of caspase-1, which cleaves the inactive cytokine pro-IL-1 beta into the active cytokine called IL-1 beta. So in the literature, until this paper, IL-1 equals inflammasome, inflammasome equals IL-1. So IL-1 is overproduced in a variety of chronic and acute inflammatory disorders. In particular, you can see in inflammatory bowel disease, in multiple sclerosis, in asthma, in inflammatory arthritis, especially rheumatoid arthritis, too much IL-1. So because IL-1 is overproduced in these diseases, the development of inhibitors of IL-1 signaling has become an important part of modern anti-inflammatory therapy. And there's one drug known as anakinra, or Kineret, which is widely used in the treatment of rheumatoid arthritis, which is a recombinant version of a natural inhibitor we have called IL-1 receptor antagonist. So we treat patients, try to lower their IL-1 beta signaling so their arthritis is better. And anakinra was approved about 15 years ago or so in clinical trials that involved about 2,500 patients. And one of the things that was present and better than TNF inhibitors, which increase the risk of tuberculosis, was what they felt a modest increase in the risk of infection. But we found there is one exception to this. And the way that we did that is by looking at the US Food and Drug Administration post-surveillance marketing for adverse events with drug therapy. And the reason we did this is because about 5 years ago, we had published a paper in Nature Immunology with Michael Caron's lab at UCSD, in which we had the observation that mice that are genetically knocked out for IL-1 receptor are more susceptible to strep infection. And I got a new postdoc named Chris LaRocque who had done his graduate work on inflammasome signaling. And I said, hey, you know, there's something about the inflammasome and IL-1, and maybe we could collaborate on that as part of your postdoc. He did the bacterial stiff arm story as his main project. But this project has turned out probably to be more interesting because of his innovation. So when we looked at those adverse event reporting system, we did note that the risk of invasive infection was not particularly high, with one exception. There is a many hundred-fold increased risk of severe invasive group A strep infections in patients receiving anakinra. And this turns out to be most associated with these severe flesh-eating infections. And this was not present with inhibitors of other inflammatory pathways that are used in rheumatoid arthritis. And what we did in the lab is found, in fact, that if you treat mice with anakinra, you can inhibit IL-1 signaling, but they become more susceptible to group A strep infection. In fact, if you just mix macrophages with group A strep, the macrophages try to kill the group A strep, but if you put anakinra to block IL-1 signaling, they are worse at killing the strep, just like an IL-1 receptor knockout macrophage would be. So IL-1 beta is produced in response to group A strep infection and is important in defense against group A strep infection. But here was the fascinating result that was totally unexpected. We started using mice that are missing caspase 1-11. These mice have no inflammasomes of any kind and are basically felt to be unable to generate IL-1. And that was true with certain bacteria like group B strep, group C strep, staph. When we knocked out all inflammasomes and infected macrophages with group A strep, they still made IL-1. They still made IL-1 even though they didn't have an inflammasome. So what was going on? Well, we went further and used a newly published inhibitor of NLRP3, another component of the inflammasome, to see what effect that would have on signaling. And we found that it blocked inflammasome activation by multiple different stimuli, but in the case of group A strep, you still had IL-1 beta being produced, even though you knocked out the NLRP3 inflammasome. But because the IL-1 beta was still being produced, the animal was still resistant to group A strep infection. We figured that group A strep must be making its own caspase, that processes the inactive cytokine into the active cytokine. And by mutating all the different proteases that the bacteria makes, we discovered it was Spv, that one protease that happens to be turned off during the pathogenesis of invasive disease. And you can see here that it's responsible for the IL-1 beta maturation. And if you just clone Spv into immune cells, they'll start making a bunch of mature IL-1 beta. We developed an assay where we compared the total proteolytic activity of different proteins with their capacity to generate functional IL-1 signaling, and we found that Spv groups over here with the known activators of IL-1, such as caspase. And what we discovered is that group A streptococcal protease cleaves caspase-1 actually 11 residues upstream of where, cleaves IL-1 beta, pro-IL-1 beta, 11 residues upstream of the normal caspase cleavage site, producing a functional signaling molecule. This bacterial caspase action also provokes the pyroptotic cell death of macrophages, which is a hallmark of IL-1 beta activation and inflammasome activation. And an interesting thing is that Spv is associated with the adverse effect of anakinra on immune defense against the pathogen, because you have eliminate Spv, you no longer see a therapeutic effect of anakinra. So Spv has been known to cleave just about anything. It's a very active and broad spectrum protease. It cleaves pretty much every host defense factor. It was in fact reported once to cleave IL-1 beta. They mapped the cleavage site incorrectly, but it was nevertheless reported. Plus it cleaves all of the group A strep virulence factors. Remember that it's turned off during the development of invasive disease. We actually had a theory for why it was turned off, which involved, well, if it's degrading a lot of its own virulence factors, it has to turn itself off in order that these virulence factors were expressed. Here you see Spv from group A strep degrading all the other virulence factors, but when you pass the bacteria in the animal, it turns off Spv and the other virulence factors can be expressed, and only the Spv negative bacteria are spreading through the body. We even had a Nature Medicine paper with this theory. We said, well, it's degrading all the virulence factors, including the DNase, and in order for the bacteria to escape the DNA nets, it has to turn off the protease so it can degrade the nets and spread through the body. And we found that, in fact, in the absence of the DNase, that the bacteria never underwent this shift. If, in fact, you eliminate the Spv, the pathogen is no longer detected by the immune system because it is not generating the IL-1 that activates the immune system. And now we think we know the reason why this is happening. The bacteria doesn't want to provoke an immune response, so it turns off its protease that is stimulating the maturation of IL-1. And you can see here that the switching to turn off the protease, if you're treating a patient with anakinra to block IL-1 signaling, they no longer need to turn off the protease because the IL-1 is being generated, but there's no immune response because the signaling is blocked. So what is happening in these patients is they're getting an invasive infection with group A strep, but they don't have to turn off the protease because the immune system is no longer detecting it with IL-1. So the reason they're dying when they're getting anakinra and are unfortunate enough to get strep deep in their body is because it's chewing them up from the inside. So the basic discovery here is that while everybody thought this is the way that IL-1 is generated in the immune system, we have discovered that there is an independent pathway of direct activation by bacterial proteases with group A strep. And we've now found that this is also true with a few other pathogens, including Pseudomonas. And interestingly, evolutionarily, pro-IL-1 and IL-1 receptor go all the way back to zebrafish. But the caspase cleavage with the canonical site in inflammasomes is only a mammalian phenomenon. So what we believe here is that in the ancient immune system, you have pro-IL-1 beta signaling actually arose to detect bacterial proteases. So if you're invaded with a bacteria that's making a broad spectrum protease that could cleave all your critical factors, you need to respond to that. So what we do is we create a protease trap, a protease detector, which is an inactive cytokine that has a hot spot for protease cleavage, and then you release a signaling cytokine. And that's been there, and it's still there as part of our innate immune system, but it was never recognized because we focused on the highly evolved immune system of mammals. And what you see here is that the mammals have said, why do we have to wait until we're being chewed up by the protease if we can recognize the pattern recognition molecules of the host as a signal of bacteria, then we can get a head start on the process. And that's exactly what a canary in a coal mine is. This, in fact, is the simplest innate immune system ever described. So the accompanying commentary of this article said that a single protein that is both the sensor and effector of innate immunity without involving anything else in the process, which is really interesting. So why would you make a protease that activates the immune system? Why would a bacteria make something that stimulates a protective immune response against it? Well, this comes back to the final thing. Remember, all that research was on invasive group A strep infections and, systemic disease. Well, what is really more common is strep throat. 600 million cases a year in the world. And that's probably where all the evolution is going on with this organism. It wants to jump in somebody's throat, stay there for a while, and then move on to somebody else's throat. And if you've ever seen strep throat, I'm a pediatrician, you say, tell the patient to say, ah. You look in there, and what you see are fiery red tonsils and a lot of pus. There's a lot of inflammation. And remember that group A strep is highly resistant to killing by neutrophils and pus. But what is not resistant to killing by neutrophils and pus is the normal flora of your mouth. So group A strep has a different strategy when it's in the throat. It doesn't want to hide from the immune system. It doesn't want to have its big capsule. It is creating fire, inflammation. Its name is Streptococcus pyogenes, pus-generating Streptococcus. So what it's doing is it's flying around from patient to patient and causing inflammation to eliminate all its competitors and take over your tonsil. And then you'll feel miserable and drooly and spread it to the next unsuspecting victim, like your schoolmate or little brother. And we found that if you eliminate the protease that is triggering this inflammation and do a tonsil infection model in the mouse, we call it nasal associated lymphoid tissue, it can no longer cause infection. And if you give anakinra, you cannot establish strep throat because you no longer get this inflammation. So you could actually prevent strep throat with anakinra, but at a very small risk of getting a life-threatening flesh-eating infection that spreads through your body. So it's kind of what I would call the dumpster fire strategy of infection that is happening here. So thank you for your attention. Sorry for the technical glitches. And this is our lab again in San Diego, and happy to take any questions if there's remaining time. Pippin, are we running late? Shall we move on? Or do yes.
Clinical significance of this higher susceptibility when you treat rheumatoid arthritis?
Right. So the clinical significance is that these patients are getting flesh-eating strep infections at an alarming rate. So a number of different, certainly doctors that are treating patients with anakinra need to be aware of this risk. Another concept is that they could be given prophylactic penicillin, just like we give patients with rheumatic fever to prevent recurrent strep infections. But it also suggests that drugs that are now in development as inhibitors of the inflammasome that block NLRP3 or block caspase-1, because they still allow the function of IL-1 in detecting bacterial proteases to be intact, that those could be therapeutic drugs that would inhibit IL-1 beta-driven inflammation in whatever inflammatory disease you're trying, but without the increased risk of infection. So those are the main clinical implications. The other clinical implication is that maybe if IL-1 beta signaling was blocked, you could treat strep throat and prevent people from getting, the painful or recurrent strep throat. But we demonstrated that experimentally here by using anakinra and inhibiting immune activation. But there's probably a smarter way to do it. Can anybody think of that? It'd be to inhibit the bacterial protease that is processing the IL-1. And in fact, I've enlisted one of our upcoming speakers, Dr. Perry, to work with us and use his skills in modeling protein-drug interactions for inhibitors of this protease that could be deployed as a treatment, an adjunctive treatment for strep throat. We'll see how it works. Excuse me?
The PMS, post-marketing surveillance,
Yes.
could not have been done on the recombinant protein because recombinant proteins are only about 10 years old.
Yeah, so no, it's basically, we're looking at it came on the market 13 years ago.
Yeah.
And then and the post-marketing surveillance that we're studying is really the last 8-year window. And it's a relative reporting risk. It's not an absolute, risk, because doctors have to feel motivated to report the infection. It's not mandatory reporting. So we're just comparing it to all other drugs in the database. Compared to all other drugs in the database, the increased risk of invasive infections is actually 333-fold in the final analysis, which is pretty bad, I think.
Excuse me, sir, just a question.
Yes.
I was just wondering, do we know exactly how SPV is actually regulated?
Yes, it's interesting. So it is not produced very much during the logarithmic phase growth of the bacteria, and then it is strongly upregulated in the stationary phase of the bacteria. And then selective mutations in the COV-RS system, which is often a negative inhibitory system, actually lead to the downregulation of expression of Spv. So a key regulator is a 2-component system, very much like the one you described yesterday, the SAERS system in Staph aureus, with their histidine kinase and a sensor that regulates Spv expression. And then there's a single standalone, we call it RALP3-like regulatory protein that also is involved in Spv regulation. So Spv is produced both under culture conditions and in vivo. Seems mainly to be growth phase regulated rather than, other environmental cues like iron or things like that, but then can be turned off by mutation in either of the 2 regulatory systems I mentioned. Okay. Thank you.